So that was a public real-world database that was across multiple institutions to look at clinical outcomes for patients with KRAS G12C mutant non-small cell lung cancer, but looking at other standard of care therapies that are currently approved, including chemotherapy, chemoimmunotherapy, and immunotherapy by itself. So for that presentation, we took a candidate approach looking at frequently co-mutated genes in KRAS G12C mutant non-small cell lung cancer, such as p53, STK11, and KEAP1. And what we observed is that high-risk population of patients with KEAP1 co-mutations was also found to have poor outcomes, so lower progression-free survival, lower overall survival compared to their wild-type counterparts, and a similar signal for STK11. We didn't observe a significant difference for p53 co-mutations. Then when we dissected that further across different treatment strategies, what we observed is that chemoimmunotherapy was associated with worse clinical outcomes when in the presence of a KEAP1 co-occurring alteration. Well, we also observed that similar findings for patients that were treated with immunotherapy alone. But quite surprisingly, I think one of the interesting findings there is that for platinum doublet chemotherapy alone, STK11 co-mutations did not seem to have a significant impact, both in progression-free survival and overall survival. This is something that has been previously shown through by other groups and by other research efforts, but it was still an important piece of information, especially as we have multiple efforts and multiple inhibitors that are starting to make their way through clinical development, with some already being planned for a frontline treatment strategy at this point. So we hope that this is an effort that will help guide some of these novel clinical trials that are currently under development or being planned at this time.